Okay. Well, Dr. Boly, thank you so much for agreeing to sit with me. And yeah, it's
Pleasure.
been a very interesting experience to learn from you. Can you give us a brief introduction and tell us a bit about yourself to start off?
Sure. So I'm a neurologist, actually. My specialty is now epilepsy, and I'm doing 25% clinical time, 75% research. I'm at the University of Wisconsin-Madison. I did my PhD with Stephen Norris in Belgium at the Coma Science Group. Then I moved here actually, kind of after doing a postdoc with Carl Friston too for a year. I moved here to kind of settle down here and to combine neuroimaging work in patients and in controls with integrated information theory as a framework for consciousness. So my main topic is basically consciousness and its
Right.
disorders. Yeah, and my main way to look at it is
Mhm.
basically through neuroimaging, intracranial recordings broadly. Yeah.
Yeah. Thank you. Consciousness is like such an interesting topic, and like there's so many different ways to define it. So when I ask, like, how do you personally define consciousness?
Yeah, I think most people who are in the consciousness field would agree that what consciousness science is after is trying to find an objective explanation for subjective experience.
Hmm.
So consciousness being defined at what it feels like, phenomenology, what it feels like to be, a person. Basically, like if you're unconscious, nobody's there, yeah. And if you're conscious, then you can have so many different experiences. But it's basically what disappears during dreamless sleep. Yeah. And then there are different ways to approach it, but there's I would say, a good consensus now that what conscious science is after is
Mm,
we try to explain this subjective feeling of, being. Yeah.
right. Okay. I like that very much. Yeah. So diving a little bit deeper into the methodology itself. So you've used PET, fMRI, like TMS, EEG, and like intracranial recordings as well to study consciousness. How do you choose like the right modality or a combination for a specific research question?
Right. So, well, you have kind of two ways to go after consciousness science that actually are very complementary. One is kind of more data driven, what we call the neural correlates of consciousness. But
Right.
it's more than a correlation. You actually look at different conditions and try to find a common signature across
Right.
lots of conditions. And then that's exploratory, and then depending on the technique you use, you may, try to see what you find. And then what I like to do is combine this with more like a top down, more like mechanistic understanding attempt, let's say,
Hmm.
kind of you have so many data, so many modalities, you try to find an explanation for the data that is broad. That is
Hmm.
basically, you have two different types of questions and in consciousness. One is what I was saying, like the difference between something and nothing, like being
Right.
conscious of anything as a state. So that's one kind of global change, and you look at that in anesthesia and sleep and coma, and then the other one is once you're conscious, so your brain already satisfies properties of being conscious. And then you look at what it feels like for like different types of experiences, being spatial, being categories, being, faces, being, but basically it depends. You need to have a framework in consciousness that can explain both. Can
Right.
explain both why your consciousness is not, and also can explain why you have the experience you have right now at this moment, depending on your brain activity patterns, and why, different brain regions would be involved in different contents and stuff like that. So I like to kind of have this explanatory framework to also test, to kind of then find some new predictions that are interesting that we can test and understand more about brain activity in that experience consciousness. They are very complementary. So depending on your experimental question, especially in the theory part, then you actually need to have a critical assessment of the modality you want to choose. Yeah.
Right.
And they are pro and cons. The reason I'm broad is because there's not one perfect technique. Yeah. Functional MRI, of course, has whole brain coverage and high spatial resolution, but it's also slow. The time course of consciousness is more like in the hundreds of milliseconds.
Right.
And so it's nice to actually, that's kind of what we did also with Xuqian Melanie. She was, I think, keynote speaker last year in these experiments, for example, where we test theoretical predictions with the cogitate. It's in present nature now. We're excited about that, data set because it's actually a way to probe theory predictions with the advantages of different modalities like functional MRI versus intracranial recordings. So depending on the question you have, the modality is gonna be critically assessed to provide some kind of information. Yeah.
Yeah. Yeah, that makes sense. Yeah, the question drives like everything else, I suppose, and
Right.
bidirectional, right? Yeah, that makes
Drives your
sense.
experimental design and
Yeah.
it drives your choice of technique. And yeah, it's a great plus actually that we have all these different ways to look at the human brain. Because for consciousness science, the gold standard of knowing if what someone else's feels is actually to ask them,
Yeah.
like with some consciousness, device skills. Yeah, but basically subjective report. And animals, you can get some report, you can get some information in some conditions like tasks, but you can't ask animals if they dream or not, You
Yeah.
can't ask animals easily like how they imagine something. So, I mean, data are really much more limited than in humans. So these neuroimaging techniques we have and multimodal are actually a great window.
Perfect. Amazing. So then multimodal imaging has like the potential to like bridge the spatial and like temporal resolution gaps. How has this like, integration like shaped your recent findings of the dynamics of consciousness?
So, well, we use a combination of techniques, and I could take maybe the example of sleep because that's one topic where we actually use these different techniques in parallel. So we did, for example, high density TEG studies with 256 electrodes, yeah? And then you wake up people at night and you ask them if they dream or not. Interestingly, in the vast majority of sleep, actually, we're dreaming, but we just don't remember in the morning. And so sleep is more like a state of unresponsiveness, but there's a lot going on.
Right.
But you
Yeah.
can compare epochs where you dream or not, and then find what makes the difference in the brain for what makes the most different, what is the most predictive, you know, feature for being conscious versus not as a state. Yeah. And so we used high-density TEG first, and then we saw that interestingly, when you look within sleep, dreaming versus not dreaming, you find that the most robust signature, both in non-REM sleep and REM sleep, which are very different states, but it's actually mostly in this posterior hot zone, occipital, parietal, temporal area, kind of posterior central cortex. But that's high-density TEG. It's great because it's kind of fast. Temporal resolution is great, but then spatial resolution is not so great. Yeah. And so then you want to complement that. You want to do some functional MRI studies. You want to do some intracranial EEG studies, and we're doing both right now. And so functional MRI, what's exciting with it is we have a PNAS paper published like 2 years ago where we show that you can actually track slow waves. The EEG slow waves are a great predictor for being conscious versus not, or inactivation locally, if you want. We actually have a signature in the spectral power of the Bull signal that actually has that great spatial resolution now. Yeah. And so we can go and wake up people in their functional MRI, and ask them, were you dreaming or not? And do the same with more like spatial resolution. Yeah. But then it's a marker, and direct marker of slow waves. Then we also can do intracranial recordings. You don't have whole brain coverage, but you can also confirm and validate things. So basically, I do like to stay multimodal.
Yeah.
And
Yeah.
single neuron recordings, I'm establishing that now too. So I do like to have these different windows combined with a framework that can really tailor predictions to modalities. Say, with that modality, I'm more confident in that particular finding versus another one.
Yeah.
To me, it remains the way to go.
Yeah. Yeah, that makes sense. That makes sense. But like, these techniques are like, continuously evolving. Are there like emerging neuroimaging tools that you're particularly excited about? Do you think that might be transformative for consciousness research? I don't know, in like the next 5 years or so?
Again, I do think a combination of technique is likely to continue, at least in humans, yeah?
Yeah.
To continue to provide the richest answers. Now, I'm excited about additional studies, neuroimaging studies that are looking more into like the mesoscale. Actually, and it's because our framework, integrated information theory, we have predictions that a certain kind of local connectivity really is important for consciousness, like this dense nearest neighbor, and then the hierarchical connectivity you find in posterior parts of the brain. We call it pyramid of grids. You have this kind of map-like, nearest neighbor connectivity in a lot of sensory areas, and then this hierarchical connections between them that are really like a strong mesh. But it's like mostly this local connectivity we think is really important for a mix of differentiation and also like causal relations, in the network. Anyway, we have this prediction about this mesoscale connectivity. And I do think both this kind of progressing layer of MRI and or like really these mesoscale functional MRI studies, these patterns. And then also like work like Nico Westcott is doing with this kind of histological MRI for the anatomy, progress in that, I think it's actually can really inform even more detailed predictions about what to find for
Mhm.
consciousness in the brain. Another thing that I really am excited about is combining neuroimaging with neuromodulation.
Mhm.
And so, for example, there's a tool we're starting to play with called temporal interference, which sounds really promising to me because you can actually stimulate wherever you want in the brain. And we're starting to kind of look at how it could change your contents of consciousness. And, as an epileptologist, I do a lot of direct electrical stimulation for mapping brain function and experience in patients, but it's again kind of limited. And if you have these non-invasive neuromodulation tools that progress, and maybe we can also perturb, brain activity and look at effects on consciousness and combined with neuroimaging because you can do fMRI on that. I think that's also really exciting. Yeah.
That's exciting. I can see
Kind
that.
of more resolution in the functional MRI, spatial resolution in a way, yeah. And then also like the anatomy in the human brain, and then this kind of combining neuroimaging with perturbation approaches.
Yeah. Like the age-old question of like structure function, that relationship as well, a little bit, like bridging that.
Yeah, we know that anatomy matters, but anatomy is not enough. I mean, the same brain, if it's deep asleep versus awake, can be conscious
Yeah,
with conscious, yeah.
right. Yeah.
But you kind of need both, really. Again, like there's been a lot of interest in neuroimaging and these large scale networks, but actually for our predictions for consciousness are also a lot in at that local level. Both matter, yeah, but there's
Mhm.
a lot to explore and understand as well with the new tools. Yeah.
Makes sense. Makes sense. You mentioned integrated information theory as a central framework in your work. How do you operationalize such an abstract theory into like testable neuroimaging hypotheses?
Yeah. So, it's been tested and continues to be tested in different ways. And that's the advantage of being a kind of at the root of the theories, like really discussing with the theory group here, like, okay, so what do we predict in different situations? So one modality, for example, that has been used and continues to be used to test the theory is transcranial magnetic
Yeah.
stimulation
Mhm.
combined with what we do in IIT, or integrated information theory, or IIT, we actually look at properties of consciousness. Like, if you study memory, you look at properties of memory. Well,
Yeah.
we look at properties of consciousness,
Right.
and then we try to see if the brain can make it, basically. And so there's like consciousness is unified, yeah, it's integrated. You have only like one experience at a time, with everything bound together. And then
Mhm.
so it's like there's a lot of differentiation in there, lots of structure, So we have this prediction that, well, if your consciousness is like this, then the brain that supports it should also be integrated and differentiated. Causal complexity. That has been a way, to test the theory, and that has been quite productive. I've been involved in my past life also in Belgium, and up to now, also I'm bringing it in the ICU with an R1 with MGH, and here we're bringing that complexity measure in the ICU to try to use it to detect recovery of consciousness and predict recovery of consciousness in the ICU. It's
Nice.
been debated as like basically the best measure we have for finding out if someone is conscious or not.
Yeah.
Yeah. So
Uh-huh.
this is like one application where you kind of look at the resolution. We use EEG because that's more the resolution of, conscious experience, like 100 millisecond, and then you manipulate causally and then you see what you find. Now there are some other predictions now, and I'm also very excited about that, about the contents of consciousness. And that's where, for example, you have these adversarial collaborations that have been used to test IIT. There's one with the global workspace, the Cogitate framework that Lucia Meloni was talking about last year at OHBM. And then now there's another one also that it's called Intrepid. It's comparing basically prediction of predictive coding and free energy principle compared to IIT. Yeah. And
Mhm.
there, depending on what is actually nice with these adversarial collaborations is you have like different opinions of what you're going to find with the same data. And so it's kind of more interesting even than us, predicting. But it has been used, for example, we predict in posterior cortical areas should actually be the maximum of decoding, but also like if you actually add some other areas in the brain, your decoding is kind of maxed out because that's where the substrate of consciousness is. From there, we kind of predicted that there would be basically high gamma activity in some areas of the brain that would track the stimulus duration in especially these posterior cortical areas, and that's kind of refined, or like some other synchrony predictions that have to continue to be tested more. But I mean, there are like a few different predictions that you can already derive from the data. The more precise we are about the contents of consciousness in IIT and what it predicts, the more we can also push the predictions further. Yeah. And so we have actually a program now we started with conscious and unconscious as the foundation, for finding, properties of that a brain should satisfy to be conscious at all. But now we have a program to try to explain spatial experiences and where they are in the brain, and then why time feels like flowing and how it could work, or like these categories. Like when you see a face, you have the category, but also like the particular face that you see right now, it binds the general and the particular. So we're kind of really trying to explain the structure of experience and try to map it to the brain and come to more predictions. And so I'm excited about that, but because I think that's really new. For example, we have a prediction that spatial experiences that depend on this kind of grid-like connectivity in visual or modality specific areas,
Yeah.
Even if the cortex is silent, like, when you have a blank screen, or like there's a part of the cortex that is not stimulated at all, it should actually matter for space. And if you have a lesion in there, space should shrink and stuff like this, that actually you would not necessarily expect or think about. And then the theory helps to find some new ideas of how to be challenged,
Nice. Yeah. Okay, fair enough. But then having used these in like clinical populations, have you found them to either support or challenge like the IITs and the predictions that you might have had?
It's quite interesting, this story. I mean, I did my PhD on coma, right? But I quickly moved to not only study coma, but also sleep and anesthesia and now seizures, because what happens with patients that cannot communicate with you is basically you cannot really know if the person is there or not. If they cannot remember subjective experience, well, our gold standard is subjective report. So these patients in vegetative state and minimally conscious state, a priori, you don't know. You have to infer from your best marker, like, TMS, EEG, PCI is our best marker, if they are there or not. Now, interestingly, if you look at the TMS EEG results, you see that the patients in minimally conscious state, as we call it, the ones that show some signs of purposeful behavior, they actually have systematically high complexity, like 95% of them have high complexity. While in the majority of patients that are unresponsive, like 81% in vegetative state, they are predicted to be unconscious by the best marker we have, which is PCI. It works always. When people can tell you I'm conscious or not, PCI always works, basically.
Okay.
You can trust that as your best marker available, at least for now. But so what is interesting is there seems to be a difference between these two populations. We find 20% of patients in the vegetative state that are conscious. So that's still, but then, at the same time, we see that if you do have response to command and even like just visual tracking, purposeful behavior, properly, there is, as far as we know, like a huge likelihood of having someone conscious there. Now that you know that, you can compare patient population.
Right.
What makes the difference between the two? One is largely conscious, the other one is majority unconscious. When you look at the difference between these patients, the areas that are most different between the ones that are conscious and not are actually posterior central areas. A lot of sensory areas are the most predictive. And that fits with our prediction of what we find also during sleep or seizures, that the areas that are most predictive for being conscious seems to be these kind of more posterior central areas. But overall, the findings in these patient populations, also, by the way, the findings for prognosis of who's going to recover from anoxia or TBI, it's mostly consistent with that. And it's actually also something quite interesting to try to explain, the contrast we see between these posterior areas and these prefrontal areas. And so we do think it's really related to anatomy. They are like macaque paper and marmoset, that suggests that the posterior part of the brain and the prefrontal are very differently organized. I was saying posterior areas is that dense mesh of local connectivity, with a lot of overlap. And then in the front is much more like modular locally, not so much local communication, and then more diffuse, projections.
Right.
It's kind of really cool finding within the cortex to try to kind of challenge, is it like in more experiment to see what is the role? Is there any role, maybe in some tasks, not, of prefrontal areas, and then kind of try to find or confront different explanation for why it would make a difference. We say it's because consciousness is differentiated and integrated, and you need this kind of a mix of differentiation and connectivity, all together to make it. But I do think it's a very interesting observation that comes from a lot of these clinical studies, as well as the studies that control, consciousness when you discard other compounds like responsiveness, and you really look at conscious versus unconscious more like this way. I think it's just a very interesting thing to explain. Yeah.
Yeah. That's exciting, actually. Here at OHBM, reproducibility and open science are like, key themes. How do you approach reproducibility in such a complex multimodal experiments?
Well, that's something where we do have here in Madison an interesting setup where we have animal studies and then we have multimodal,
Mm-hmm.
matching studies. And we really to try to test our predictions and our hypothesis for the sleep part too. We have some theory of sleep, but in this multimodal way. So basically, for example, the role of different cortical areas, also with activity or perturbation,
Yeah.
we look both in animals and in humans. In humans, we look across modality, functional MRI, intracranial EEG, and high-intensity EEG. And then we also look across conditions. So we look across sleep, anesthesia, seizures, patients. So it's really an attempt to validate not only in one condition, but also across modality to challenge it and kind of try to get confidence.
Makes sense. Okay, so then the next notable question is like lab to clinical practice. Like, what are the translational steps that you think are required to make the sleep, bring consciousness metrics from the lab to a clinical practice, especially like in critical care or like epilepsy units?
I briefly mentioned to you, I'm very excited because we have
Yeah.
that protocol where we're trying to bring TMS and VG in the ICU.
Yeah.
And there actually we have a plan is like two parallel routes. So TMS-EG right now is like 60 channel, like the heavy TMS. And I'm doing that. I go in the ICU myself right now. We just started with Brian Edlow, at MGH.R1, where we want to validate that TMS can provide an accurate marker of consciousness in the ICU in these patients that are responding to command, or even like tracking like minimally conscious patient. We want to see the results we get there. We also want to explore at least the first try, who's going to recover. If you find a complex response, if it predicts more chances of recovery. So I'm doing this actually with kind of the heavy setup that, worked with 60 channels. Now cross fingers, we have a pending, also like smaller grant within IH to try to develop a portable machine with less electrodes that we can use more routinely. So, but we're really trying to push this also because TMS is a super causal way, TMS-EG, very high signal to noise ratio. We're really trying to push that in the intensive care. And then we're also comparing it with other modalities. So we have some functional MRI also in the study. We have some the high intensity EEG, like resting state. And we're going to try to find some other measures, and see how they compare. Also, I was involved in designing that task. Imagine playing tennis in the functional MRI with Adrian Owen. So we're also doing imagine playing tennis.
Nice. Okay.
So we're kind of comparing in the ICU all these different markers and see one how they're good to detect covered consciousness, and then or like even like subtle consciousness, like this minimally conscious state, and then the other one is to see how patients recover. I'm also involved in doing a lot of studies in epileptic seizures, because that's one of the huge problems, like maybe one of the biggest problems with patients with seizures is that can come anytime and you completely lose control, you lose consciousness, you have these grand mal seizures where you can die, and we still don't fully understand how it's going on. And so I started to study more these states, and you have different forms, like sometimes it's cortical slowing, sometimes it's huge trauma of activity and you're not there. And so there's a lot to understand to try to help the patient to try to prevent it, hopefully with neuromodulation we can help them if we see the networks and the patterns involved. But also for consciousness science is very interesting because seizures is the only condition that I know where you have this kind of huge increase in brain activity, everything like super synchronous, and then bum, you disappear. And so, yeah, basically the hope is to go not only in ICU, but also help patients with epilepsy and try to improve clinical outcomes if we can understand how the seizures can progress there.
Yeah, makes sense. Okay. Switching gears towards neuroimaging specifically too, like you mentioned briefly that earlier, like neuroimaging can help you move towards like mechanistic predictions. What's your vision for like the future of consciousness research at like the population neuroscience level? Do you think we can scale like individualized metrics of awareness?
I do think conscious science is making a lot of exciting progress also in the method, trying to dissociate consciousness from consequences and try to find which cognitive functions are actually associated one-to-one with consciousness versus the ones that actually may be more consequences, like say working memory, you can have a conscious or unconscious working memory, things like that. So understanding better the links between consciousness and cognition in the view of improved and revised consciousness science methods, I think is going to be very interesting. In terms of individualized measures, there's a lot in consciousness science that is relevant clinically or for well-being. After all, well-being is how you feel, right? So the more we understand the parts of the brain, the kind of mechanism for consciousness, and also like the quality, not only this kind of spatial temporal aspects, which are important, but the more complex ones, like, the sense of self, the structure of sense of self, how it actually relates, binds to emotions, or like, all this, we are not there yet, but the more we can actually piece the whole story together.
Yeah.
And also link how you feel to what you do, then I think the implications are going to be quite broad. So beyond neurology, which, is my own topic, but also I think this kind of computational neuropsychiatry linking consciousness and cognitive sciences together. To me, I think it's becoming more and more feasible to go in that direction together. And I think it's really exciting.
Nice. Yeah. Okay. I'm curious about this dissociation between consciousness and cognition. How do you guys go about doing that?
There's an ongoing debate.
Okay.
And basically, it depends how you ask questions to. A lot of studies still ask all or non-binary questions. Did you see something or not? But in a lot of situations, consciousness has been demonstrated to be graded. So if you ask subjects to kind of design their own scale, you see that it's typically not all or none. But sometimes you see something very clearly. Sometimes you had no idea something was on the screen. Sometimes you have a glimpse of awareness. You're not really sure what you saw. And if you're not aware at all that there was anything on the screen, you are changed behaviorally. And when you start to see something, then you are both changed. Okay. But at the same time, consciousness is very rich. We experience tons of different, features at the same time. We only do something about a minority of them. Yeah.
Right. Yeah.
You can dissociate consciousness and cognition in that sense that it's not because you saw something that you're going to do something about it. Then, like, you're dreaming or meditating or whatever, you can have that dissociation. And that's the one where we're getting better and better at doing, like, task irrelevant or no report paradigms. That one is actually used more and more to carve out consciousness from these consequences. Now, again, the other dissociation is debated and it's an interesting topic, but you have to see how the questions were asked to go there. At the same time, it seems to be that, at least of what I think, there's something true of what they distinguish in neuropsychology, this kind of crystallized versus flexible cognition. Yes. This kind of knowledge of the world, like the categories, the faces, the spatial aspect. This is actually kind of something that we have in our conscious experience. But then things like, I don't know, speaking or working memory, I know what I want to say, but I have no idea how I say it. So it's quite likely that some consequences, say carried out by basal ganglia or like prefrontal cortex, can be dissociated truly from experience. And there are kind of different ways, like consciousness and cognition can be linked, if that makes sense. So typically I would say what we do is a consequence of how we feel, but there can be some exceptions. But overall, if you have no idea if stimulus is there on the screen, you're going to get changed.
Right.
Or minimum, but what we do typically is a consequence of what we feel. But how exactly cognition and consciousness interplay is still a quite interesting question to be addressed. And again, because there's so much progress in cognitive science and in consciousness science, I think that's going to be a very exciting topic for the next decades.
How does this like manifest in the brain? Like, what are the neural markers of engagement in like consciousness versus cognition?
Well, you can have a little bit of a flavor of it. When we do these experiments, when we contrast task-related kind of target detection versus task irrelevant, And then if you look at contents, you actually kind of need to zoom on one particular content at a time, like say the cognitive experiment, or like one aspect of this content, like orientation versus favorite. But if you actually look at what is the signature for that content, consequences, actually going across tasks, like doing both the task relevant and the task irrelevant and see what's common across them, then you can really appreciate, and there's a lot, the differences that you can see in different task contexts. So a lot of frontoparietal activity that you see, basically, if you look in neuroscience database, and you see at what is activated in response to stimuli, frontoparietal areas, if you go that forward inference, kind of this classical meta-analysis, they kind of show in every contrast, But then if you look at what's content specific, then it's a different answer typically. So it's like really, you need to kind of be broad and look across condition and see what is most consistent, talking about reproducibility. Yeah.
That makes perfect sense. So just had one last question for you. What advice do you have for early career researchers at OHBM who want to tackle like complex, high-dimensional questions like consciousness?
If I look at my own experience, I do think it's really important to know your methods. Even if you're a user like an MD like me. So I am so grateful Carl hosted me in his group for a year, so I was feeling more confident of like, the methods part. As a kind of end user, you still really need to know what you're doing in the like, you can't know all the codes. It's always a teamwork, yeah. And to me, I've been educated by Christophe Phillips. Maybe you met him. He's a wonderful engineer in Belgium. Anyway, he was always saying to me, before you come and ask me a question, Melanie, you need to make your homework. You need to know 2/3 of the stuff, and then maybe then we can have a good conversation. But I would say that's really important to go to methods courses to try to be really educated in what you do in the methods, and also the advantages and limitation of the techniques, because ultimately it's your responsibility what you do. Yeah, I would say if you want to study consciousness, it's a broad topic. I mean, it's like saying you want to study cognition. There's so many ways. I mean, you can be interested, and it's good to be interested broadly, to know the literature, to converse with a lot of, different researchers, but it's also important to find a team of people you can work with so that you can combine this. That's why I moved to Madison, Wisconsin, yeah, to work with Thovinakere, Chiarelli, and the gang, because then I can actually do my own experiments, but keep that broad view. And then you have your own niche, of your subfield, but keeping the communication and really kind of thinking together about how to explain the data in a comprehensive way. See what I mean? Within the big picture. I do think that's important. Yeah. And then, if you're interested in integrated information theory, you can always reach out to me. We have also a school that we do like every 2 years. And if you're interested in that, we have a wiki and things, and we're trying to make our stuff, we know it's complex, but more understandable. And the reason why it's kind of seemingly complex is also because consciousness requires a different kind of framework. It's very different from typical science, where you need to look at consciousness from the inside. You need to look at the properties of experience. And then we frame it in terms of structure, with these distinctions and how they bind together. It's a very different way to look at, at the brain too, because you need to take this view from the inside, and it's just a very different mindset in a way. So, I mean, it's good to be broad. It's good to know all the different approaches, so you can also have your critical mind about it. If you want to learn from us or like give us ideas on how to challenge stuff, we're very happy to get in touch. And yeah.
As a graduate trainee, I appreciate these insights very, very much. So thank you so very much. Dr. Boly, that's all the questions I had for you. I'm looking forward to your keynote talk at OHBM 2025 in Brisbane later in the summer. It's always such a pleasure to meet and learn so much about the neuroscience. I think our OHBM audience will truly appreciate.
I'm looking forward to seeing you, and I'm really honored to be able to speak with the HBM crowd about our work on consciousness. I really think the future is very exciting, and the more links we can have together and brainstorm, and what to do together, that's wonderful. So thank you.
Yeah. Thank you so much. Okay, have a wonderful day.
Bye-bye.